Good morning. I'm going to talk about two species of midge that are being observed in soybeans and one in our state and one in other states, and then a quick forecast for 2021. So the first insect is the soybean gall midge, and we'll quickly go over its life cycle. Let's start with the overwintering larval cocoon in the soil. And then next spring, it'll go through a pupal stage, and then the adult will emerge once the temperatures warm up. In Nebraska, it's typically about mid-June. The adults are very short-lived, about 3 to 5 days. The female will lay eggs on the stem or openings or wounds in the stem, and then the egg hatches into larvae. There's 3 instar stages or growth stages, and the larvae feed on the soybean plant for 2 to 3 weeks. The early instars are clear or white in color, whereas the more mature larvae are orange to reddish color. And we don't really know how many generations we would have here in North Dakota, but I expect we would have at least 2 generations per year. So the cycle would continue until fall. Okay, the larvae do the damage to the plant. You may see out in the field a wilting plant, or you may see a dark discoloration at the base of the plant stem, or dead plants even if the infestation is severe. If you take your fingernail and peel back the dark discoloration underneath the epidermis, you'll see the larvae feeding on the stem. This is just a close-up. You can see a white young larvae and then the more mature orange-colored larvae. The field symptoms, you may see a dead row of soybeans near the field edge, and they may snap off easily, and there may be some swollen stems near the base, near the soil line, that are called galls. This group of insect in the Cediomyidae family are gall midges. So we know that soybean gall midge is highest in the field edges. So we do see a reduced plant stand near the edge, but as you walk into the field 100 feet or more, the plant stand usually increases, but it's still below the average plant stand that you would like. And they do impact yield. All those small dead plants won't produce anything, and then you'll get reduced pods and seed. And of course, in the edge, we've seen 100% field loss. And as you get into the field 100 feet or more, it's about 20% yield loss. And this data is from Nebraska, where they're really having a problem with this insect pest. So this is an economic insect pest of soybean. And they're doing a lot of research in the infested states. And crop rotation with a non-host, corn or wheat, is one of them. And we do see reduced number of soybean gall midge cocoons in fields that practice crop rotation. At the Ithaca site, they had continuous soybean there, so no crop rotation, and it had the highest number of midge cocoons, 99. Whereas the other sites, Eagle, Waverly, and Fremont, had a much significantly lower number of total midge cocoons. So crop rotation is one way we can manage this pest. And then late planting date has been shown to have a lower incidence of soybean gall midge infestation than earlier planting dates in early May. However, they repeated this trial this past year and they found the reverse was true. So more research definitely needs to be done. Chemical control, they looked at insecticide seed treatments and they did not get any efficacy over the overwintering generation. Foliar insecticides that were timed for emergence of the adult midge again, they saw marginal efficacy, and they tested a wide variety of different classes of insecticides. So there may be something out there that looks more promising, and more research is needed. We did do a regional multi-state survey for the soybean gall midge, and all the states that have yellow dots is where it's infested or known to occur, and all the states with blue dots have no detection of this midge species. And here's the distribution of where it's at currently as of 2020. 5 states are infested in 114 counties, and it continues to spread and increase each year. And of course, we're concerned in North Dakota with the Transverse County, where it's right next to Richland County in southeast North Dakota. Midges in general are weak flyers, but we don't know how fly it could move with wind. And we've been surveying for it for the last 2 years, thanks to the North Dakota Soybean Council. In 2019, we focused in the southeast region. In 2020, we utilized our IPM scouts, and we also did a focus survey more in the southeast. And fortunately, we did not find it anywhere. But we did have several complaints in those starred counties, is where growers called to let me know through their agent that they have larvae, orange larvae in their stems of soybeans. So we went out and we collected the larvae and we DNA typed them, and they were not soybean gall midge, but the white mold gall midge, which is the second midge I'll speak about. So no gall midge yet in North Dakota. And it is easy to confuse it with the other species I'll talk about, the white mold gall midge, Karshomyia caulicola. This one feeds on the fungus, white mold fungus, Sclerotinia. And so you can find it in other crops than soybean, dry beans, canola, potatoes, sunflower, and even poppy flowers. This one feeds on the mycelium growth of the Sclerotinia fungus. So it doesn't cause economic damage to the soybean like the soybean gall midge does. But as you can see, the larvae are orange color, so it is easy to confuse it with the soybean gall midge. And we did survey for this white mold gall midge for 2 years, and we found it fairly widespread in southeast North Dakota and Minnesota as well, wherever we found Sclerotinia. So if you look at the adult midge, the white mold gall midge is uniformly gray antenna, legs, and wings, whereas the soybean gall midge has white and black banding on the legs, and the wings are mottled. But again, you're probably not going to see the adult midge. They're very short-lived, only 3 to 5 days. You're more likely to find the larvae in the stem. And with the white mold, the color is white to orange, and the soybean gall midge is also white to orange, and many entomologists say it's a darker orange, a more reddish orange. But again, they're difficult to ID based on color. You need to get them under a microscope and look at the little knobs on the posterior end, and then it takes a taxonomist to do that. Or we can do DNA typing to identify the species. So they also differ with their location in the field. The white mold gall midge is usually found later in the season after flowering and the onset of white mold, whereas the soybean gall midge can be observed as early as the 3rd leaf stage in Nebraska, where there's severe infestations. And in the field, you can find the white mold gall midge wherever there is white mold. It'll be either in the stem or in pods that get white mold. Whereas the soybean gall midge is concentrated more on the field edges, and especially will be near the field edge closest to the previous year's soybean field. that's infested. And then they're usually always near the base of the plant, near the soil line and under the epidermis. So what to do if you see a white orange midge larvae in the stem of your soybeans? Please collect 10 to 20 larvae and then put them into a glass vial with rubbing alcohol or ethanol. You can get these at your county extension office. Label with your county, date, GPS coordinate, and contact information. Send them either through your agent or directly to me, and we'll get the DNA testing done to determine its species identification. So the forecast for the upcoming year does not look good. A severe drought in about 60% of the area of the state. So 2 insects I'd like you to keep your eyes out for when you're scouting is grasshoppers. And as you can see, the past 2 years, we've had increasing populations as indicated by the yellow square and the red triangle. So we use the threatening rating as our action threshold. It's based on the number of grasshoppers per square yard in either the margin or field. And they're hard to count when there's hot out and there's a lot of grasshoppers and moving every which way. So buy yourself a nice, inexpensive sweep net. They're only about $40 or less, and they last you 10 years or more. And 4 180-degree sweeps is equal to a square yard. So then you don't have to do the counting. I recommend doing at least 20 sweeps to get a more accurate count and then divide by 5. So there's many different insecticides registered in grasshoppers. Dimlin is the insect growth regulator at the top there. So you need to get that sprayed on early when they're in the nymph stage. And it is safer for other beneficial insects and birds, etc. And then there's organophosphates, there's premixes, and then the pyrethroids, the 3 A's, and then Stewart. But grasshoppers typically come into the field late, so be sure you check the pre-harvest interval, and each chemistry is a little bit different. We did do a grasshopper insecticide trial on soybeans this last year at Castleton, and we tested many different pyrethroids, both at the low and high rate, and then 1 premix cobalt advance at the low and high rate. And the untreated check is in the red square over on the right-hand side. And you can see that we had significantly lower grasshoppers per square yard, yellow bar, and percent defoliation, the blue bar, in all of the insecticide seed treatments compared to the untreated check. Likewise, we had the highest yield in all of the insecticide seed treatments compared to the untreated check was much lower, usually about 10 bushels per acre lower. And so there was no differences between the different insecticides. So you can use whatever you would like. Both low and high rate were effective. We had a combination of late nymphs and adults present at the site. The other one I'd like you to watch for is the 2-spotted spider mite. They're very tiny, yellow with 2 dark spots on the sides of the abdomen. So buy yourself either a head-mounted magnifying lens or a hand lens, or you can use the white sheet of paper method where you wrap the foliage over the sheet of paper and you'll see the tiny mites moving slowly. But you're more likely to see the damage symptoms first. Leaf stippling, yellowing and bronzing of the leaf, and mite webbing. If you get to the mite webbing stage, it's a severe infestation. This is how they move into the field. They spin strands of silk and they get blown on these silk webs into the field. So we got several factors that are favoring spider mites. The drought stressed plants are favored for mite infestation. Low rainfall, that means fewer diseases that naturally control the mites. Hot temperatures, fast reproduction. 5 days is all it takes for them to complete their life cycle. And the natural enemies, there's not as many of them because they like cooler temperatures than 90, usually in the 70s and 80s. So scouting is very important. They usually move in from the field edge, but make sure you get out there and walk that U pattern into the whole field. I encourage you to watch the Scouting for Spider Mites and Soybean YouTube video that's been up for a while on the web, and that will go into more detail on scouting. So the susceptible stages are early flowering through past seed, or for past full seed, when the mites are most economic. There's no count. You do kind of a general rating or for your spray threshold, and you look at the plant, and you want to see heavy stippling in the lower leaves and some into the middle canopy. You don't want much going on in that upper canopy where the pods are. And this is the action or the spray threshold. And again, mites start from the bottom and they move their way up the plant. And they're also found on the underside of the leaf. So here's a list of the insecticides and miticides for spider mites. Again, you got the pyrethroids 3A and organophosphates 1B and premixes. And then at the bottom in orange is some miticides that we have. And again, the premixes vary, so check that because each chemistry is different. And the mites typically do move in late in the season is when we see them. And again, we need to spray. You need to use a good amount of water volume. Remember, those mites are at the bottom of the plant and underside of the leaf, and that's why we need to use adequate water volume. And they're most severe during drought, as I mentioned, so use a high labeled rate rather than a low rate, and a adjuvant to make sure that insecticide droplet is getting down to the plant. Then avoid pyrethroids. They flare spider mite populations. The only active ingredient we can use is bifenthrin. Then all of our insecticides do not control the egg stage, so we need to go back, re-scout the field 5 to 7 days to make sure we're not having a resurgence of spider mites. Then alternate modes of action, of course, to prevent insecticide resistance. Minnesota has documented Lorsban organophosphate resistance. And we don't want that in Minnesota. So here's some new publications that we have available. The soybean gall midge publication and white mold gall midge is new, and the common arthropods of soybean in North Dakota is meant for your truck. It's made with that heavy poly indestructible paper, so it can get wet, and it's to help you identify the different pests of soybean. And that's all I had, and I think we'll be taking questions later.